I'm Mona Flores, and I'm excited to share with you why I love to study plants. Plants make ideal study systems for understanding how the environment and environmental change affect an organism's ability to function and survive. Especially because plants don't move, all of their strategies for growth and reproduction involve modifications and adaptations to their anatomy, physiology, and morphology, including the ability to quickly respond to changes in their environment via a phenomenon called phenotypic plasticity. Now, I also study how plants respond to these environmental changes across developmental stages. And it's important because at different stages of a plant's life, a plant may be able to respond differently to environmental changes, and plasticity itself can change at different stages of a plant's life. Now, I study a specific organism called Puakala, or Argemone glauca, which is an endemic Hawaiian prickly poppy distributed on all Hawaiian islands. It produces these beautiful and conspicuous defense traits, including waxy leaves, prickly vegetative tissue, and exudes this bright yellow latex, which may be toxic. It also produces a very pretty papery white flower. Now, like I said, it's distributed on all the islands. It's very versatile in different habitats. It grows in dry areas as well as on coastal rocky shores and on lava rocks, making this a really versatile organism for studying. I use these three stages to understand how environmental conditions might affect specific features or traits in the Puakala, and I do this in a greenhouse setting by manipulating conditions like water and light using shade cages. Now, I use all of this fancy equipment to study the plant. I'm measuring its traits like photosynthesis, chlorophyll, the electron transport rate, which tells us how efficient photosynthesis might potentially be under certain conditions. I also want to know about its latex, how much does it produce and how toxic it could be. And we take a look at how leaves change in response to specific conditions. Now, one of my favorite things to learn about plants are these different strategies that they use. In Puakala, I'm excited to learn how at different stages, whether a seedling, a juvenile, or a vegetative mature, changes their ability to respond to these conditions. I'm especially excited about expanding this experiment and taking it into a field setting where I can study an additional stage in the Puakala, the reproductive stage, which will allow me later on to use seed germination and viability as a proxy for fitness to establish whether there is a fitness advantage to these different strategies and trait changes at different stages of the Puakala's ontogeny. I'd like to thank ARCS Foundation Honolulu for supporting me in my research.